It's fine. I'm glad. So I was thinking of staying until Sunday, but then I was like, nah. Like I'm taking care of my cat and like my two cats, and I was really glad that I didn't because like it got extremely icy and terrible yesterday. Oh my god. Yeah, me too. I kept like I guess I haven't checked to be completely honest. I never get campus notifications for that. I don't know, like maybe I have them turned off for that class or something. Like the answer. Well, yeah, that too. I definitely am cold. I want to know so I can know how hard it is to take the last exam. I will not be sleeping for the next but yeah. It was that or there's like a new and both of them yeah, both of them are only offered or no that the one that was only offered spring and like directly released and it's also like planning on doing so might be yeah. It may be pretty cool. Maybe. I don't really know what to expect from them. I might end up asking the professor who teaches that if she'll be on my committee as well. I need to have a project.
Yeah, that too.
Oh god, last week, someone in a different lab, one of the PhD students in a different lab, there was like this big email chain with like 5 people in it, and she mentioned, or no, my PI like volunteered me, to help her with some stuff, because like I helped with some other preliminary stuff before that, and he was like, yeah, I thought Nicole would be easy, or like what her schedule is, and you guys can like work on this like I would like that. And I mean she's pretty cool. But she told me she was like, yeah, or like in the email she was like, yeah, I'll get together with her so soon so she can have all this information for her thesis. And I went, for my what? Excuse me? What information were you told? I really hope she goes surgery tonight.
Okay, welcome back. Glad you people showed up. Always easier to talk to living people rather than something on a computer screen. I'm just going to start right in here, I guess. So, when we last met, I was starting to introduce you to this phenomenon called variable gene expression or gene silencing or things like this. This was a phenomenon that only really became apparent when people started introducing foreign genes into multicellular cellular eukaryotes and allowing them to integrate through non-homologous recombination. And what they found, as you recall, is not every transgenic behaved like every other one. So in like the case, this is related to one that I showed you, but different from the one that I showed you in an earlier lecture. When people were taking, for example, the genes that determine flower color in petunias and trying to either enhance the color formation or to reduce the color formation by introducing either a sense construct that would simply add a additional copy of the same enzyme the plants already have, and so maybe it would enhance the color, or by introducing an antisense version, which I'll go into in a few minutes, but it's basically the whole concept that if you could make an RNA complementary to the normal RNA that the plant makes for that particular gene, well, in theory, the two RNAs could base pair to each other, like the two strands of DNA would, and then in some way block expression. It was thought initially that, and I'll tell you again why, but it was thought initially it would probably block translation. But what they found is, so they expected two very different phenotypes, whether they introduced the antisense gene or the sense gene. But what they found was quite different. So this is the original flower in this particular case, and all of these are transformed with a sense construct. Now this is actually very similar to the patterns and differences and the variations they got when they introduced the antisense construct, which for which they thought they had an explanation. They thought, yeah, like I said, maybe it was blocking translation. Although it didn't explain why there would be flowers that were completely blocked in the expression and other flowers that were just partially blocked. But then when they introduced the sense construct, all of this became moot, because the sense construct produced the exact same phenotype. So it seems as if there is another process, unanticipated, operating in these multicellular eukaryotes that we're all familiar with. And some experiments, a number of experiments were done. A very early experiment was able to show that so first of all, here we're looking at a very complicated process. This is an example where we're looking at the effect of the expression of the transgenes complicated by the development of the flowers and when those pigments are going to be produced. So this kind of a model really didn't clarify very much. But in simpler models that were developed, where they were only introducing, for example, an antibiotic resistance gene like NPT, they could see that in a number of cases, maybe as much as 1 in 7 or 1 in 8, maybe as few as 1 in 15, that NPT gene turned off. And in these early experiments, they did look for methylation, knowing very well that methylation does inhibit the initiation of transcription. So they looked for methylation differences, and they found methylation differences that correlated with the silencing of the NPT gene. But correlations are complicated, and we're looking at a very complex phenomenon that could have multiple causes, and attributing it just to this one perhaps was, you couldn't be confident that this one simple explanation of methylation accounted for all of the examples they saw. But even if it did, they couldn't explain what triggered this methylation in 1 out of 7 or 1 out of 8 or 1 out of 15 of the transgenics. Well, one of the earliest experiments that began to give us a clue, and so what I'm going to be doing now for today and Wednesday and maybe afterwards, is I'm trying to go through some of the additional experiments that seem to indicate what was the components of this silencing process. We want to think mechanistically. Why this gene and not identical copies of this gene in other individuals? Why in this particular case, was it methylation? Because we know that methylation only affects things when the methylation occurs at the promoter region or the upstream region of the promoter. And they didn't have and there's a lot of sites there that were not assayed for methylation patterns. So is it true that the methylation pattern they saw at this one restriction site actually could account for the phenomenon they saw downstream? So there was lots of cases. But then in the case that I showed you before break, they were able to promote specific methylation of a gene if we began to see an excessive accumulation of RNA complementary to that gene. And in the case that I was using, in the example I was using, one of the first, if not the first example that people had, that gene was a viroid cDNA. So remember, these viroids are circular RNA molecules. They're normally seen as pathogenic. When people introduced single copies of the viroid cDNA, nothing happened. It just got transcribed. When they introduced concatenameric copies of the viroid cDNA, those concatenameric RNAs self-processed, because they're ribozymes after all, into circular molecules that then proliferated and at some point seemed to turn off the transcription of the copy that was integrated into the genome. And the simplest explanation for that, drawing on examples from a variety of sources, was that the circular RNAs that were formed base paired back to the complementary sequence that we put into the genome, and then recruited RNA-dependent RNA methylases to methylate first the transcribed region of the integrated gene, and then as those methylases drifted upstream and downstream, methylated the promoter region to shut off transcription of the integrated gene. Now, viroids were still being produced through their natural mechanism, which involves, rolling circle replication. But the important thing for us is that the integrated copy, the one that started all of this, was silenced. And so people began to think maybe transcriptional gene silencing is induced when a particular messenger accumulates too rapidly in the nucleus and is not exported from the nucleus, and therefore it has the ability to bind back to its template and recruit these RNA-dependent DNA methylases to shut things off. Okay, so that gave us a mechanism for specific inactivation of a certain gene. Although, we were still left with questions. Why did it happen in this case, for example, in this plant number 1, and it didn't happen in plants 2 through 8? So we had a partial explanation, we didn't have a complete explanation. But things then became even more complex when other transgenes were investigated. And so the one that I'm going to be talking about is one that was a project that was done by Fernanda de Carvalho, and it involves this topic here, suppression of the beta-1,3 glucanase transgene expression in homozygous plants. Because this is going to add some more parts to the big mystery. The purpose of the experiment was not, like all of these things, was not to investigate silencing. Nobody was looking for silencing. I think I mentioned at one point, at the time, a lot of people, including myself, thought that there were artifacts involved, that this couldn't be a real process, that somebody just misinterpreted some kind of results, and who knows? Everybody was looking for an excuse why their transgenics didn't work, and so, this is just another example of people making up excuses with very little evidence to support it. But I was wrong, and so were a number of other people, though. So in this particular case, as I said, nobody looked for silencing. In this particular case, what people did, what De Carvalho did, was clone a beta-1,3 glucanase from one particular variety of tobacco. This enzyme is a fungal defense enzyme. It chews up the walls of fungi, and so they thought if they could overproduce it, they would make plants that would be more resistant to invading fungi. It was, reasonable thing. So what they did is they took the genomic sequence in this particular case, and they cloned it behind a strong plant promoter, in this case a 35S promoter, in front of a terminator from the nopaline synthase gene. Just a terminator from the beta-glucanase gene. And then in a tDNA vector that also contained a selectable marker based on the nopaline synthase promoter, the NPT gene, and a 3' sequence from octapine synthase. And they transformed this into plants. And they generated 5 different lines. Now, each line was clearly heterozygous, or you might even say hemizygous. It had a single, as far as they knew, a single copy of the transgene in it. Now they thought, well, that's one copy of this beta-glucanase. Maybe if we made this homozygous, we would make an even more effective antifungal defense system for these plants. And so they found the one plant out of those 5 that really had the highest level beta-glucanase expression. They selfed it, and then they screened the seeds. And by taking the seeds of the self homozygote, heterozygote, and plating them on kanamycin. Now, remember, when you have a heterozygote, so that means, let's just say, one copy of the NPT gene, and itself, so you're going to generate 25% of the progeny with no copy of the NPT gene. They will have inherited the untransformed chromosome from both the male and female parents. 25% would have inherited the NPT gene and the accompanying beta-glucanase gene from both male and female gametes. And so they would be homozygous, and then 50% would be heterozygous. They would have inherited one transformed chromosome, one untransformed chromosome. So when they screened these by growing the seeds on kanamycin, all the seedlings that did not have a copy of the transgene would die. And that's what happened. So they've eradicated 25% of the population. What they then did is they just assayed for the beta-glucanase gene in the remaining progeny. And what they saw was 66%, so that means like 2/3 of the survivors, let's say. We've eradicated the untransformed plants. Now we've got to this remainder here. 2/3 of the survivors showed the amount of expression that the transformed parent showed. Transformed parent had one copy. They therefore said these 66% of the seedlings were also expressing only one copy of the transgene. They were also heterozygous, as Mendel would have predicted. The other 33% should have had, in theory, twice as much beta-glucanase, because they would have been the homozygotes. But instead, the remaining 33% showed no additional beta-glucanase expression other than that which would be normally found in a non-transformed tobacco plant. So as if the transgene had just shut off, but only shut off in the homozygous state. Everybody okay there? It's going to get hairy. Well, the next step was to say, well, look, let's just follow the developmental progress of this transgene from the moment that it emerges from the seed up until, maturity. and see if it was always off, always silenced, or was it possibly only silenced later in life. And so what they found, to cut a long story short, is they looked at the developmental progress, they looked at the expression of the beta-glucanase gene during plant development. Now, plants are kind of weird, because when we speak of development, we have to speak of it in two different contexts. First, there's the time from emergence of the seed, what we would think of from the time of birth. That's one aspect of aging for these plants. But the other aspect of aging is that within any given plant, the oldest leaves are at the bottom of the plant, and the youngest leaves are at the very top. So you've got a developmental gradient going up, as well as a developmental gradient based on time alone. And what they did is they harvested plants after doing some preliminary studies. They harvested some leaves, some plants after 5 weeks from the time they germinated, and then also after 6 weeks. And they assayed each leaf in each of the plants that they were looking at. So in some cases, they were looking at leaves 1 and 2. These are the oldest leaves. And then they were looking at leaf 3, leaf 4, leaf 5, which is the youngest leaf. And each of these two populations of plants, 5 weeks from germination, 6 weeks from germination. And they assayed those for the beta-glucanase transcript by northern analysis. Afterwards, they also did some genotyping. And this is what they found. If we simply look at the first of all, we always should look at controls. So the control here is non-transformed plants. And we see that the probe they were using didn't hybridize to anything in the non-transformed plants as expected. Then they looked at plants that later were characterized as being heterozygous. And what they saw is that as the leaves were younger, they tended to have a slight increase in expression of the beta-glucanase gene. But that pattern, going from oldest to youngest, is not too striking, and it held the same whether the plants were 5 weeks from germination or 6 weeks from germination. And then they looked at the homozygous plants. And what they saw there is that in the homozygotes, at 5 weeks, all of the leaves were expressing beta-glucanase, maybe a little low, but it was there. But by 6 weeks, the gene shut off throughout the entire plant in every leaf. So in that short period of time, something triggered silencing of this particular gene. And keep in mind, all these plants had an NPT gene as well, and that was still functioning even at 6 weeks. So we're looking at a very gene-specific silencing process, almost like the viroid story, almost. Any questions on that so far? There's so many questions that arose when this phenomenon was first found, and so a number of experiments were done. Today you might not have considered these worth doing, but at the time they were, because nobody had seen this before. So one of the questions they said is, well, we know that we're seeing silencing in plants that have gone through meiosis to make them homozygous. So is it really the homozygosity, or does it have something to do with the physical age of the plants? You know, the size of the plants, if you want. So one of the things we can do with plants, that would not be possible easily with animals, is we can regenerate clonal copies of those plants. For example, by taking the leaves from an expressing plant, putting them on medium with the proper concentrations of various hormones that trigger the formation of little shoots. And then they were able to assay the expression of those shoots, asking the question, is this silencing stable through morphogenesis, from a differentiated cell into a non-differentiated cell, back into a differentiated cell, and eventually a little plant? And what they found is that if the plants if this shoots, or let's say this, if the leaf came from a plant that was already expressing the beta-glucanase, for example, because it was a heterozygote, then all the shoots also expressed it. You didn't see silencing in some fraction of these shoots generated from the heterozygous plants. But if you took a homozygote silenced plant and generated shoots, all of those shoots were as silent for the transgene as their parent had been. So we can say that this silencing process is heritable through somatic development, at least somatic morphogenesis. The next experiment that let's see, so another experiment that they did, let me just show you that one. And this one is, was probably not as convincing as not as important in the long term as we thought. So one of the possibilities was maybe we really are producing so much of this messenger RNA that it's clogging up the nuclear pores through which messengers have to leave the nucleus to get into the cytoplasm so they can be translated. And if they're clogging nuclear pores in some way, well then we would expect other genes to show silencing at the same time. And so in order just to look at that particular question, they ran two-dimensional protein gels. So, I don't know, you've heard about two-dimensional gels, maybe in biochem or something? So the basic idea is very simple. You first run the proteins in a system that allows them to separate based on surface charges. And then you take that strip where the proteins have separated from each other based on charge, and you lay it on top of an SDS acrylamide gel, the standard kind of gel we run proteins on, which denatures proteins. And then when we run the proteins out of that strip into this denaturing gel, they separate according to size. And so by combining these two different methodologies, we can resolve thousands of proteins that otherwise would look at like bands on a normal SDS gel into spots. And if you do it right, you can probably detect more than 1,000 independent spots. If you don't do it right, you probably detect only a couple hundred. But it's enough to give us an idea of what's going on. So here we have our untransformed plants that we've run this way using this two-dimensional system. And you see these spots and streaks, which is, unfortunate, but that happens. Each spot is at least a protein that differs from its neighbors either by size or charge. When we look at the hemizygous or the heterozygous plants, so these are the ones that are expressing the beta-glucanase, although it doesn't necessarily look that way, most of these could be aligned, most of the spots here could be aligned with the spots in the untransformed plant except for this one, which is the beta-glucanase protein that we are overproducing. And it's revealing, I think, that this transgene is producing so much of that particular protein. Okay? And then we go to the homozygous plants, the ones that we couldn't detect this protein in. And we see that spot is missing. But we don't see any other spots. When people made the comparisons as carefully as they could with better gels and some of these, they couldn't see evidence that any other spot was silenced as well. So this is sort of a pre-proteomic way of investigating which proteins are being produced in each of these genotypes. And in a pre-proteomic way, the conclusion was no other protein was being affected by the overproduction of this particular one. So we don't think, or we think it's less likely the messengers are clogging up the nuclear pores and preventing the export of the messenger. Okay. At some point, they then followed through with another experiment which revealed a lot. Remember, the plant they chose was the highest expressing plant in that population they had generated initially. And when they selfed it, and so at that time, that protein was being made when it was in the heterozygous state. But when they selfed it, then we found 25% of the seeds were silenced for this gene. So they said, is this process reversible? And what they then did is they crossed the silenced plant with a non-transformed plant, and they just investigated some of the plantlets that germinated from the result of that cross. Again, we're going to look at the controls first. So in this particular case, we're looking at the, let me think for a second here what I want to say. Okay, so here we're going to look at two different crosses. And in one case, what they're doing is they're going to just cross this silenced plant with itself. And what they found is when they assayed for the beta-glucanase gene, that's GNL, and the NPT gene, which was their control, they found that all of these 7 progeny showed expression of the NPT gene, as the parent did, but no expression of the beta-glucanase gene, as the parent did. So in that case, the silencing was stable across meiosis. But when they crossed it with a non-transformed plant, they tested it and they saw here the NPT gene was being expressed actually. It doesn't look like it, I know it's very weak there. That's just an artifact of the exposure time that they used for this particular set of plants, so that everything would look kind of comparable. But in other experiments, it was clear that the NPT gene was being expressed normally in the heterozygous plants that would result from that cross. But so too was the beta-glucanase gene. So in the homozygous state, the beta-glucanase gene is silenced. If you cross it with a wild-type plant, the beta-glucanase gene is turned on. If you self that second generation to generate new homozygotes and heterozygotes, all the homozygotes again became silenced. So we are able to induce and reverse this silencing with every cross we make. Yeah.
Did they rule out the possibility that it was like deleterious to the plants just to be a homozygous due to like the morphology of the plants?
Well, they didn't detect differences in morphology, but they can't, but let's say it differently. Let's just get to the chase. The mechanism that's causing silencing is in the plant itself. Why would the plant have a mechanism for silencing genes, if not to protect itself? So although maybe morphologically there were no variations or changes in the appearance, when we think of it as a whole, when we look at it biochemically, there could have been bad things happening within the plant that could have triggered this kind of silencing. But I think the more important feature to keep in mind is all of us, animals and plants, are ready and prepared to silence any gene that seems to get expressed at an abnormally high level. And all we revealed in this case was the existence of that defensive mechanism. And as time has gone by, we've learned how that mechanism has been used in other cases. But it had to have been an early invention because it's present in all the multicellular organisms, plants or animals. Not in yeast, as far as I know, and not in. It is present in filamentous fungi. But yeast went through a lot of reductions to try to grow fast, so this may have been one of those things that said, I don't need it. No? Okay, any other questions? Yeah?
I guess I most need a reminder since we went through the break. When we learned, right, that when we were talking about the viroids,
Mm-hmm.
there was like so much RNA that it formed a trimer with the DNA, right? And then that causes certain proteins to come in and then methylate, which silences the gene.
That's right.
Okay. These experiments like before they found that information? So like that wasn't known?
So, two things. Your question is a great question. There's two aspects to it. First, in terms of when it was published or developed, I believe it was a little bit before the viroid paper was published, but they're almost contemporaneous, within a year. But when they looked for methylation, in this example, they didn't see it.
In the homozygous?
In this case where we're looking at the silencing of the beta-glucanase gene.
So the homozygous had no methylation?
Well,
That's
it,
what-
didn't have
what-
any, it had its normal methylation.
Okay. Okay.
And, but we're gonna try to patch these two concepts together. Okay, 'cause I'm not a splitter, I'm a joiner. I like to find universal principles rather than specific principles. Okay. Are there any other, but there's some other, this is a very important detail actually, because this actually does relate to what Eric was asking very strongly. After doing all of this work, they had been assaying for the RNA, which is obvious, the messenger RNA, which is obviously a cytoplasmic molecule. Well, they looked for methylation, but really couldn't find it. But you know how chancy that whole question of how do you assay for methylation of the critical sites is. So they said, we're going to bypass any additional experiments looking for restriction sites sensitive to methylation, and we're simply going to ask, was there RNA from these genes in the nucleus? Because if transcriptional silencing was operating, whether it was by methylation or some other magical process, there should have been no RNA accumulation in the nucleus, just as there was no RNA accumulation in the cytoplasm. So they did some in vitro nuclear runoff experiments. And what that basically means is you isolate nuclei from the plants that you're talking about, and you incubate them in a cocktail with a buffer and some salts and some radioactive ribonucleotides. And although there's not a lot of new initiation when the nuclei have been isolated from their cells, any messenger that was in the process of being transcribed would continue to be transcribed. But now, because you're giving it radioactive nucleotides, the messenger would be finished with the incorporation of radioactive nucleotides. And then you kind of can crush those nuclei. And in this particular instance, what they then did is, so now you've got a mixture of all of the primary messengers being made in those isolated nuclei in your test tube. What you can then do is take an aliquot of that and let it hybridize to a fixed amount of DNA on a filter. You can choose any genes you want that you know are going to be expressed, and the amount of the messenger in that nuclear extract will determine how much radioactivity binds to the particular gene you spotted on the filter. Is everybody clear there? Not so clear. You can raise your hand, you don't have to say it.
Repeat it one more time.
Sure, okay, thank you. We have made radioactive RNA by isolating nuclei and incubating them with radioisotopes like P32, alpha P32, GTP, ribo-GTP, alpha P32, ribose CTP, and so on. So that all the RNAs that were made in our test tube will be radioactive. Now we want to assay for specific genes. And so to assay for specific genes, we take a known amount of the clone copy of that gene and spot it on a filter and incubate that filter with this random mixture of nuclear RNAs. And if those nuclear RNAs are complementary to the DNA on your filter, they will stick, and then you can do an autoradiogram and visualize how much stuck, and you can put it in a scintillation counter and count how many counts were actually bound to that particular spot. In this way, you're able to quantify how much of those primary RNAs were present in the nucleus for each of the genes you want to study. Does that help a little bit? Okay. Well, as always, we're going to be looking at nuclei isolated from a homozygous plant and from the hemozygous plant. That's our heterozygous plant. Okay. And we're just going to look at the expression of a couple of genes. The first is going to be, we just put vector DNA down, a certain amount of vector DNA. This will be a control for the specificity of the hybridization. If we use, so this is like Southern analysis, if we use too relaxed conditions that have been relaxed too much, we could get non-specific binding of the the nuclear RNA to the fixed DNA. So we have to prove to people that we were using stringent conditions. And the way this was done was just by putting vector DNA there. And as you can see, there wasn't very much radioactivity bound to it. The second thing they wanted to do was they took an endogenous plant gene. So this is a protein that's highly expressed in leaves. And they incubated it, and what they saw was that indeed the homozygous plants had approximately twice the amount of the heterozygous plants. So that gene was being expressed, and being expressed, you know, pretty much, I'm sorry, I shouldn't have said it that way. Skip that. Yeah, I was wrong. What this was intended to show is that, I'm looking ahead here, what this was intended to show is that this gene, which is present in the same copies in the homozygote and in the heterozygote, had the same amount of hybridization. And there is a slight difference, but it's, you know, a statistically insignificant difference, basically. So yes, the endogenous genes were being transcribed in both parents, in both plants, the homozygous plants and the heterozygous plants. Now here's a little complicated one. The next gene they investigated was the MPT gene. They knew the gene was being expressed in both plants, but they had to verify that for themselves with this nuclear assay. And what they saw is that, well, in this particular case, for reasons that were not clear, the NPT gene was not being expressed in these hemozygous plants to the level they expected. But it was clearly being expressed. And there may be some partial silencing even going on there, we don't know, of a different kind from what we're talking about. But the important thing is there was expression of the NPT gene, and there was much more expression in the homozygous plants, where there should be 2 copies of NPT, not just 1. Okay? So far, these controls agree with our expectations. And then we get here. The hemizygous plants definitely expressed the beta-glucanase genes when they assayed for messenger levels, and as expected, they were definitely expressing that gene in the nuclei. They had to. I mean, it was made in the nucleus and shipped to the cytoplasm. So this was okay. The homozygous plants didn't have any messenger in the cytoplasm that they could detect. So the question was, would there be no messenger in the nucleus which would be consistent with transcriptional silencing? But instead, they found twice as much beta-glucanase in the nucleus as they found in the hemozygous plants. So in other words, in this case, we are not looking at a process that silences transcription. Transcription is going just fine. But something happens between the time that the messenger is made and accumulates in the nucleus and the time that it gets exported to the cytoplasm. So we don't think this can at all be explained by any kind of methylation process. So now we are clearly in the realm of having two different kinds of silencing. Post-transcriptional silencing, such as this, where the messenger is transcribed but then destroyed somewhere, and transcriptional silencing where the messenger is just never made. Any questions on that particular paper? Obviously, there's no clear explanation for the process, for the mechanism by which this happens yet. We're still going to develop that in some additional papers. But if you have questions on this, we can talk about those. Yeah.
Are there, so aside from, you know, it being clogged by nuclear pores, are there any other sort of explanations for it that have been ruled out?
Well, we ruled out the methylation, and we ruled out the nuclear pores. I think our imagination failed at that point. I mean, there's some kind of, I tried building a model once where it had to do with the location of the genes in the nucleus, that in some way, so that when they were paired, so when you had two chromosomes with complementary sequences, they would be pulled away from the nuclear membrane and therefore be harder to export the RNA. It was all sort of fluffy and based on, you know, inappropriate experiments out of Drosophila, frankly. We think they're inappropriate. So I don't think anybody had a model. Any other questions yet? Okay. So let's just go through let's just summarize some of the basic points of this experiment. First of all, the same transgene worked in most transformants, but not all transformants. That is this concept of variability in expression. People made a variety of different constructs. They said maybe this is all due to the promoter. Maybe this is all due to the absence of in this case, maybe this was all due to an intron. Maybe it was due to the absence of an intron. Maybe it was due to the 3' end. And so various constructs were made with different promoters, different introns or absence of introns, and 3' ends. And the ultimate conclusion was this kind of silencing could happen to any gene, no matter how you constructed it. It was not unique to a specific promoter or a specific intron or any of those factors. In general, it was clearly dependent on expression levels. So it really was the case that they only saw this kind of silencing in the plants making the most beta-glucanase messenger. Those others that they had isolated, which showed lesser amounts of beta-glucanase, did not show this kind of silencing when they were made homozygous. So it is definitely concentration dependent in some way. And it doesn't, and you're not, the seedlings are not born with a silenced gene. They're born with the gene being expressed, and only over time, in this particular case, 6 weeks, did the gene shut off. So again, that could be consistent with the need to accumulate a large amount of the messenger to eventually trigger a uniform response throughout the entire plant. And then also the effect we're looking at, like all of the silencing effects we are looking at in these examples, is very gene specific. Even adjacent genes are rarely affected by the process. So we're not looking at a global methylation or chromatin modification of an entire locus. We're really looking only at something affecting one gene. Long before all of these experiments were performed, we knew from studies, as it turns out, of TN10 that nature sometimes uses antisenses to regulate the expression of a given gene. In the case of, so remember TN10 is a transposon, and one of the genes that's encoded there is a transposase. And when people began to study the promoters or the transcription of the transposase, they found something a little surprising, which was that the transposase has a strong promoter that transcribes the transposase, but it also has an internal promoter called P out instead of P in that transcribes from the beginning of the gene outwards. And you'd say, well, what could possibly be the value of having that kind of a second promoter? Well, it turns out that the messenger that was made would therefore, because the messenger started within the gene, it would base pair with the 5' end of the transposase gene. And it turned out that that complement between the small P out transcript and the larger P in transcript blocked the ribosome binding site for the transposase protein. And because the regulation of the two promoters is slightly different, this allowed the transposon to modulate how much transposase it would be making in the bacteria. Because, you know, if there was a lot of transposase being made, that transposon would be jumping an awful lot, and every time it jumps, or every time it integrates, it could cause a lethal mutation. And the transposon is dependent on the viability of its host, so it doesn't want to over-mutate its host, and so it regulates the production of the transposase, not with a protein like a normal gene would be regulated, but rather with antisense RNA. And that gave us the clue that antisense RNA has some biological meaning, which wasn't clear at all prior to that. Everybody thought it was an artifact, in fact, when they assayed it. And the idea was really, like I mentioned before, simply that when you make a sense copy of a messenger and an antisense copy of the messenger, they have the ability to base pair to each other, forming a double helix, which in the case of the TN10 could block translation, because the ribosomes have a hard time breaking that up. When people realized that, they began to use antisense as a technology. Initially, the technology involved simply taking any gene you clone and then cloning it into a vector in the reverse orientation. So instead of transcribing the Watson strand, you might say, it was transcribing the Crick strand, and the plant itself would be transcribing the Watson strand. And that kind of production of antisense RNA actually was useful in changing phenotypes of various plants. So for example, in this particular case, by making a antisense RNA to one of the proteins necessary for red pigment formation in tomatoes, they could convert the tomato from a red fruit, like the parent had, to a yellow fruit, simply by blocking that one step with antisense RNA. In another case, they blocked the production of a hormone necessary for ripening, so that the tomatoes would be stopped in their development prematurely. Now, some of the processes would continue, and they would continue to get sweeter, and they would continue to accumulate all sorts of things, but they would be hard, and they would be yellow. But if they then took that hormone and sprayed that onto these tomatoes, that would reinitiate the developmental process, and now they would be able to complete this. And so this was one of the first types of transgenic fruits that was sold in this country. The idea being that we could harvest these tomatoes, ship them while they're still hard, and then when we're ready, just infuse the cargo cars or cargo holds with the proper hormone, and they would ripen in place at their destination. And then here's another case where, in fact, what they did is they inhibited the enzyme responsible for softening of the fruit, for making it a soft fruit. And when they inhibited that enzyme, the tomatoes, so whereas a wild type tomato would eventually, if left on the plant, would eventually fall off of the plant and crush itself because it had just gotten so soft it couldn't hold onto the stem. Whereas these transgenic potatoes that were unable to produce the enzyme responsible for that softening would just sit there and get sweeter and sweeter, but they would still remain hard. And I believe these are still in use for, tomato paste and ketchup. It gives you a better quality because you can harvest them when you're ready, you don't have to harvest them at a specific time and so on. Okay, so antisense proved to be a valuable tool, and we'll come back to that later as a way of regulating gene expression. But right now we want to just focus on the question of, well, we know how antisense works in TN10. It blocks the 5' end of the messenger to block, we think, the ribosome. Is that the way it works in eukaryotes? Because after all, the eukaryotic ribosome is quite different from the prokaryotic ribosome. Eukaryotic messengers are quite different. But we should prove at least that antisense works the same way. So, to test whether antisense works the same in plants, for example, as it does in bacteria, they took a gene for chalcone synthase. So this is one of these enzymes that makes, one of these genes that makes an enzyme for pigment formation. And they cut it into two halves, the beginning part of the gene and the end part of the gene. I've labeled the beginning part of the gene with a blue bar and the end part with a red bar. And they clone each of these two halves behind a strong promoter and in front of a terminator in an antisense configuration. And they introduced each of these constructs into plants to verify whether the antisense to the 5' end of the gene worked better than the antisense to the 3' end. But it didn't. Oh, I'm sorry.
Can I assume that they tried using the whole gene but antisense?
Yes, so they definitely used the whole gene, and they did see silencing. I should point out that I've known a number of people who have had problems when they used the entire gene. So actually using the entire gene is not as efficient as silencing a gene than using just a part of it.
Okay.
And it's probably because the messenger, when you have a long messenger, it has secondary structure, and that blocks its ability to bind to the endogenous messenger. Okay? So in this particular case, yeah, they knew that the whole gene would work, and now they're just going to test each half. And what they found is that, so what they're going to be looking for is a change in the color of the flowers associated with silencing via antisense RNA. And they'll also be looking for variegation and some other things. And what they found is none of the plants transformed with antisense RNA targeting the 5' end of the gene showed silencing, showed a reduction in the chalcone synthase messenger. But most of the plants generated with the antisense of the 3' end of the gene showed silencing. So this is totally 180 degrees different from what we see in bacteria. In bacteria, We believe antisense works best when it blocks translation. In eukaryotes, it seems, targeting the 3' end of the gene is the best strategy to achieve silence. So now we've got another mystery we have to explain. And since this is all supposedly about this whole phenomenon of gene silencing, we're going to have to integrate the results and the hypotheses based on this experiment with whatever we conclude from the previous experiments that I've talked to you about. So it's getting harder and harder to come up with a unified model, you think, that's going to explain all these different aspects of and mechanisms by which silencing seems to be occurring. Okay. Let me just introduce you to some terms. I think in your notes I may introduce this later, but if I introduce this now, this list, and it is somewhere in this lecture, it'll make it easier for me to talk because I won't have to watch my words so much, okay? So let me just introduce you to some basic terms that have accumulated over time with regards to this topic of gene silencing. First is an epigenetic effect, which we've talked about before. Epigenetic changes are any changes that change the phenotype of an organism without changing the genotype. Epigenetic changes are generally not inheritable through meiosis, but they are inheritable through mitosis. Okay? And we now would call most epigenetic changes, except for some interesting exceptions, the result of modifications of either the DNA or the chromatin of the gene, around the gene itself. Gene silencing, we now realize, is a general term composed of two halves. One half is transcriptional silencing, and one half is post-transcriptional silencing. Co-suppression is a term I haven't used very much. Remember some of those flowers I showed you earlier on? We had introduced a sense copy of the gene, and the entire flower was white, or in some cases, there were patches of white. What that meant is not only that the gene we put in there had gotten silenced, but the endogenous gene had as well. We call the ability of the endogenous gene to be silenced by a foreign gene co-suppression. Is that okay? Does everybody understand that concept? In other words, those plants would have, you can imagine, had 3 copies of the gene. 2 we put in, maybe, or I mean 2 that, 1 that we put in, and 2 that the plants had originally. And in the end, not only was our copy that we had constructed silenced, but so too was the endogenous copies. Otherwise, we would not have seen a white flower. We would have still had pigment produced. We're going to be talking also about siRNAs. siRNAs are small inhibitory RNAs. I'll tell you where some of the concepts behind siRNA came about, maybe a little bit today, but certainly on Wednesday. But it is the mechanism by which we believe a lot of gene silencing at least gets initiated. It is also a tool we can use to silence genes that we want to see silenced. And the process of using siRNA is called RNAi, or RNA inhibition. So the tool is siRNAs, the process is RNAi. And the fact that this process even works, as we'll describe, means that the plants already must have had some kind of mechanism in operation to generate or maintain small inhibitory RNAs. And we've learned over time that this natural use of these things involves what are called microRNAs. This is one of the tools that plants or animals use to regulate gene expression without resorting to new proteins. So they can turn a gene, they can reduce the expression of a gene by making a natural RNA complementary to the gene we're trying to downplay, or downregulate. And that natural RNA is called a microRNA. Some of these microRNAs do block translation. Some of these microRNAs will involve the actual process that we're going to be talking about, which involves the degradation of the RNAs themselves. All right. Anybody have any questions on these terms? Yeah, sorry.
When you talked about co-suppression,
Yeah.
does co-suppression only happen as a result of introduced like sense or antisense RNA, or can it happen in other methods as well?
Could you repeat the last half of that? What was that?
So co-suppression,
Yeah.
can it be, or is it only a result of introduced like antisense or sense RNAs, or can it also be from a different method?
It's mostly visible when we put in our own genes. I think for all practical purposes, we should just think of it as being limited to silencing induced by foreign genes.
Okay, so would the difference then between co-suppression and RNAi be that RNAi is specifically with like the RNAs, but co-suppression is with introduced genes, just in general?
Let me think of that follows, yes. I've tried to define it slightly differently. I've said that RNAi is the process. I know it gets confusing because different people have taken different definitions for all of this stuff. So for me, to try to partition out everything so that we have one word, one concept here, for the course at least. RNAi is the process by which we use siRNAs to turn a gene off, or by which, I think by which we use siRNAs. And then when we do this in a way, so for example, let me see if this doesn't confuse you enough. We can control gene expression by introducing siRNA, as I'll talk about in a little while. It's been a common tool in medical technology as well as in developmental biology and all sorts of genetic investigations. In this case, we're only specifically injecting a in vitro made complementary RNA or a gene that would transcribe that RNA. When it causes silencing, it could cause silencing of the gene of every gene that has that complementary sequence. So I wouldn't actually call it co-suppression in that case, because the siRNA is still going to be made, even in the silent state, because it's really different from the gene that's being shut down, if that makes any sense.
Okay, so in co-suppression,
We're putting an exact copy of the gene we want to shut off into the organism.
Okay.
And letting that gene get shut off that we were targeting, as well as the gene that produced the agent that caused the silencing. Okay? I don't know if that's going to help. Sometimes seeing more examples is more useful than trying to break it down. Because like I say, what you're going to find is that there's a lot of loose terminology out there. Some people have kind of an imperfect image of the complexity of the situation, and so they've broadened their definitions and they've misused words, and it's now become pretty common to have misused words like this. So you have to be flexible as you hear these people or you read these people's work, because you may have a precise idea, but their idea is a lot fuzzier. It's like a promoter, definition of a promoter. Which can be very fuzzy. Okay, let me just introduce you to the test organism again that we're going to, I'll see how far I can go here. At the time that all of these studies were done in plants and in vertebrates and so on, and tissue culture of vertebrates, there was other experiments going on in other organisms, and probably for the same purpose that some of these experiments that I've described were being performed. And one of the great organisms that they worked with was called C. elegans, a nematode, tiny little nematode. I think I've talked about these little guys before. They are a great genetic system for numerous processes. They have revealed enormous amount of information on the causes of aging and the genes responsible, on the mechanisms of cell death and the genes responsible, on development principles in general, in broad terms at least, and certainly on neuromuscular proteins involved in neuromuscular transmission, absolutely essential role there. They're very simple animals. They have a fixed number of cells, unlike a vertebrate, for example, so we know exactly how many cells are present in the adult animal. Most nematodes are hermaphroditic. That means that they make both sperm and eggs, and then they go through a self-fertilization process. They can lay about 200 eggs in their lifetime, which is very short, about 10 days or so. And they have a pretty basic sensory system, but most importantly, they can be easily cultured on petri dishes seeded with E. coli. So they're just great investigative organisms. At one point, when people were trying to improve antisense technology, so now when I'm speaking of antisense technology, I'm talking about the synthesis of small siRNAs in a chemical reaction that would then be introduced into the cells of interest in order to shut down a specific gene through whatever mechanism antisense was working, which again we'll get to a little bit later. But they knew it wasn't perfect. They knew that it didn't always shut things down. So in this particular case, what Andy Fire was doing was he wanted to see what were the proper amounts and conditions for shutting down an endogenous gene using antisense RNA. And the gene that he chose as a marker was a gene called UNC22. This UNC stands for uncoordinated. The gene was identified because when it was mutated, the animals couldn't swim properly. So, and there was a whole lot of genes that contributed to this, all involved in neuromuscular transmission or in muscle protein development. And so all of them came to be called uncoordinated. And what he wanted to do was to try to use this as an assay. The idea was if you took an adult animal, you micro-inject a buffer into it, for example, and you put the worm over here. In a certain period of time, that worm will crawl over here. Okay, so that tells you that the injection had no negative effects. Now, the point that Fire wanted to make was, what if I infuse that buffer with antisense RNA? If it functions correctly, it will inhibit the UNC22 protein during development, and as a result, the animal will be paralyzed. So when I put the worm here, it won't be able to crawl here, and that will tell me that I've silenced the UNC22 gene. I'm going to stop because the next part is kind of critical, and I don't want to leave it hanging. So, yeah.
Just curious, did they use any I don't know, if they had pheromones or something to cause it to go in one direction? Because potentially it can go anywhere.
Oh, yeah. And in fact, when I'm speaking in this way, I'm simplifying it. If you take the petri dish and you put a circle of bacteria out here, and you put the worms in the middle, they will just crawl away from the place where you laid them down in the direction of the food. and it doesn't really matter which direction you use.
Right.
They do have some pheromones, but mostly the pheromones they have cause aggregation of the worms rather than attraction. But food is a really good stimulus.
Okay.
These guys like to eat. Yeah.
Can you remind us of the final date and time for the final on this course?
I can't. I don't know. I don't yet. Do you know?
Wednesday from 3 to 5.
Thank you.
Wednesday what?
From 3 to 5.
Yeah. Okay. I never look. You know, I leave it up to the university to publish it. We don't have any choice in the matter. By we, I mean teachers. Any other critical features to talk about? Gives me so few days to breathe. Okay, so we're going to continue with this story and develop this part of this discussion of silencing a little bit further on Wednesday.
I got an email this morning saying, we accepted your co-op application, so you should be getting an email soon.
I don't know if anybody wanted to get a quiz, I mean a